Welcome to this short lecture on cell adaptation. In this particular lecture, we're going to look at how cells respond to stresses placed upon them to keep you alive. So when your cells are confronted with stresses placed upon them, which will impact their structure and their function, cells need to respond to these stresses to essentially keep the cell alive. Now, this particular diagram here is a schematic diagram to show you how cells are constantly exposed to different environments and how cells need to respond to keep homeostasis going and to keep the cells, the tissue, and ultimately you alive. So here we've got a diagram of a normal cell. So this cell was trying to keep its homeostasis, but what we can see is there's some arrows pointing down to cell injury, and over here would be stresses placed upon the cell. So if certain stresses on the cell, such as workload or demands on the cell, hormones, a change in pH, a change in nutrition, exposure to chemicals or irritants or inflammation, the cell needs to adapt to this particular environment. And if it can adapt, it will ultimately go back to its normal homeostatic state. Similarly, the cell can be also exposed to potentially very harmful things, such as mechanical trauma, chemical trauma, physical trauma, a drop in or a reduction in oxygen, and this is going to lead to the cell to become injured. Now, the cell needs to recover after injury, and it can do this through ways of wound repair or regeneration, such as resolution, regeneration, replication. And sometimes it needs to replace the cell type, and this is what we call a reversible injury. So this can reverse and go back to the normal cell homeostasis. But if the cell can't deal with the injury that's placed upon it, it will essentially go into an irreversible state and the cell will die. Now, Mike has done this in a video, and this is known, two types of cell death is known as necrosis and apoptosis. So Mike has done that video, so we're not going to focus too much on that. What we're really going to focus on is how the cell adapts to stresses placed upon it. So the cells can usually go through 4 or 5 main types of adaptation. These are known as atrophy, hypertrophy, hyperplasia, metaplasia, and dysplasia. Now, the first 3 I've drawn on the board. We've got atrophy here, we've got hypertrophy, and we've got hyperplasia. Now, I'm going to do that in a second, but there's a few points just to be aware of. Firstly, when you think about all the cells in your body, they are generally categorized into three ways in how their constant activity is essentially situated. So some cells are what we call labile cells, which are constantly replicating and copying themselves. This is going through mitosis. So they're very mitotically active. Then you have a group of quiescent cells or stable cells. So these cells are almost asleep. So they aren't copying themselves, aren't replicating themselves, but they can if need be. And then finally we have permanent cells, which are cells that will never go through mitosis again. They can never replicate. They can't ever copy themselves or go through mitosis to repair themselves. Examples of these type of cells would be muscle cells or neuronal cells. Okay, so that's important to be aware of because that will dictate the way that adaptation progresses. So essentially, before we get to adaptation, I just want you to really understand this diagram, that a normal cell, when stresses are placed upon it, the cell needs to adapt to these stresses to keep the cell alive. Okay, some of the exposures could be very injurious agents, so that could be, say, a reduction in oxygen, or some can be just an increase in demand. So imagine this cell is a muscle cell in your heart. If you have a restriction in blood flow to the heart, such as angina or potentially a myocardial infarction, the cell isn't getting any oxygen anymore. So it needs to respond to that injury. Hopefully it can rectify by getting oxygen back to it. But if it doesn't get the oxygen, it will essentially go into irreversible state and die. Now, if it gets injured a little bit, the muscle can repair itself, but it generally will change its state and become more scar tissue, more collagen. So this is a replacement in cell. So it's reversible, but it may not be functionally that great. But another example of in the heart muscle cell would be if you put a lot of stress on it, either through hypertension, high blood pressure, or you work it hard through exercise. The stress placed upon the heart muscle is the demand, and so it has to respond to that. And because it's not a labile cell, instead of getting increase in number, all it can do is get bigger, and that's called hypertrophy. And so now we're going to do this adaptation, at least for the first case, with these three types. Let's start over here with atrophy. So atrophy, what you can see, atrophy basically means a reduction in size. So here we can see 4 normal sized cells sitting on a basement membrane with a nice blue nucleus. Now, going down here, what we can see, these 4 cells have reduced in size. So this is known as atrophy. So basically what's causing the atrophy is a reduction in work demands or possible stimulation through hormones or growth factors. Now, the reason why it would decrease in size is it would make the cell more efficient. So if your cells weren't being used, so this is a good example of why cells would become atrophied, is disuse. For example, if you were to break your arm and you were to put your arm in a cast, you're not now moving your hand and fingers and so forth. So the muscles in your forearm aren't being used anymore. So when you take that cast off in 8 weeks, your muscles in your forearm have become smaller, which is atrophy. Now the reasons why it's doing that, well, it doesn't have the demand placed upon it, so the cells basically reduce their size, which makes the cells more oxygen and energy efficient, and it's better in the long run for everything. So that's an example. Other examples of atrophy could be denervation, so the nerve going to the muscles could be cut, and that would cause atrophy. A reduction in nutrition, a reduction in hormonal stimulation, a reduction in blood flow. So the way that atrophy can be categorized is in 2 ways. Either physiological atrophy, which is a normal response to a stimulus, or a pathophysiological response, which is more disease-like. An example of a physiological atrophy would be, let's say, the muscles in the uterus after pregnancy. So during pregnancy, all the hormones that has caused the uterus to get bigger, to grow with the developing fetus, once the fetus has passed out, the hormone levels drop, so that means the uterus drops back to its original size, and that's atrophying. Now, it's important to note that atrophy is a decrease in cell size, not in number, so these are usually in stable cells or permanent cells, such as muscle cells. Another example of, well, let's go to a pathophysiological example. A good example, pathophysiological example of atrophy would be ischemia. So ischemia, so this is a reduction in blood flow. So if you were to have, let's say, peripheral vascular disease, so this is a reduction in blood vessel diameter into your legs, your muscles in your legs would actually atrophy because it's not getting enough adequate blood supply. So that's an example of a pathophysiological cause. Another example would be deinnervation. So if you, let's say the muscles to your forearm, if you had a median nerve cut, so if your median nerve was cut in your arm, that means the nerve signals going to your muscles in your forearm being reduced or actually completely stopped. So that means all the neurotransmitters that go to the muscle, which are like growth factors, aren't there anymore, and the muscles in the forearm will actually get smaller. That's an example of atrophy. So I think that will do for this particular one. Moving across to hypertrophy. So what we can see here, these 4 cells in a normal state, but comparing to these four are much bigger. Again, like in atrophy, we're talking about cells that are permanent. So a good way to remember the hypertrophy are in muscle cells in the body. So this could be skeletal muscles and cardiac muscle cells. So usually the stimulus for hypertrophy to become apparent is usually in response to mechanical force or stretch placed upon it, such as muscle contraction, hormonal stimulation, or growth factor stimulation. So a good first example is getting bigger by going to the gym. So if you were to go to the gym and lift weights, what you're doing is you're placing mechanical stress on the muscles lifting the weights, such as doing dumbbell curls. So the biceps are being, the demands placed on your biceps, this is the stressor, is increased. So the muscles respond to this by increasing the genes or the gene expression to make more proteins for contraction. And also organelles will be increased as well, such as mitochondria, because you need more energy, etc. Another example of a physiological response is the uterus in pregnancy. So in this case, it's going to be a hormonal stimulation, not so much a mechanical force. So the hormones are going to go down to the uterus, the smooth muscles of the uterus, and tell the uterus to get bigger and more powerful. So when it comes to the point where we need to push that baby out, you have a much more greater amount of muscle tissue to do that particular act. Another example, which could be a combination of physiological and pathophysiological, could be the bladder. So if the bladder had a restricted flow, such as in a prostate, an enlarged prostate, so when the bladder's trying to empty and push the urine out, it's got a restricted flow, so the bladder needs to get increased strength, and the way it does that is also build up its muscle content within the wall of the bladder, and this would probably occur through a similar example of the gym where you place increased force and demand on it. Some examples for a pathophysiological, so a more disease state hypertrophy, would be in the heart. So this would be cardiac myopathies. So you could have an increased heart muscle size in athletes. So this is a good thing. And so what they propose here would be, as the muscle gets bigger, proportionally the length and the width increases proportionally. So as the muscle of the heart actually gets bigger and bigger, it continues to be efficient. But in some of the myopathy cases, such as dilated cardiac myopathy, what happens is the muscle gets bigger, but it gets longer in length proportional to the width. And that, at some state, the heart becomes inefficient and ineffective, and there becomes a problem with contraction. Another example of a myopathy is hypertrophied cardiac myopathy, where you get a proportional width versus length. So the muscle in the heart becomes really bulky, but then it restricts the ability of the heart to pump or fill, and this becomes a disease example. So these are important to note, that hypertrophy, like atrophy, can be physiological but also pathophysiological. But it's important to note that these two cell types are usually permanent cell types, such as muscle. Okay, and the way that it overcomes the demands placed upon it is it increases the size of the tissue by increases the size of the cell. Whereas if we go across to here, so this was called hypertrophy. Coming across to here, this one is hyperplasia. Okay, now if you were looking at the actual tissue level, in either case here, the tissue size would increase. But the difference is the tissue size increases with hypertrophy by the increase in size, but in this one, the increase in organ size is actually through increased cell number. So hyperplasia is increase in cell number, not cell size. So the cell type in hyperplasia is actually labile cells or labile or stable cells. So they're not impermanent cells. So examples would be epithelial cells, hematopoietic stem cells or in the bone marrow, liver cells, glandular cells, intestinal cells. So in this case, there's stress placed upon it, similar to here, but the way that the cells respond, these labile or stable cells, is to increase the number. Okay, again, like the other two, there's two categories of hyperplasia. There's physiological, which is normal, or pathophysiological, which is disease-like. Some examples of physiological, the inducing agent would be a hormone, changes in hormone. So a good example would be hyperplasia of the breast tissue, both going through puberty for the female or going through pregnancy for the female. So the change in hormone concentration will cause breast tissue to increase its number, such as ductal cells, to increase the ability to produce milk. A good example of a change in the stable cell would be in the liver. So the liver is composed of hepatocytes. The hepatocytes are usually dormant or quiescent, so they're kind of asleep. But if you were to cut the liver in half and remove that half of the liver out, what would happen is the hepatocytes would become activated. So the genes that get turned on for hyperplasia are genes for the cell cycle, unlike for hypertrophy, which are genes to produce organelles and genes to produce muscle proteins for contraction. So these are genes for cell cycle. So you now turn on the cell cycle and you increase mitosis. So you start replicating and replicating. So it's important to note that this has to happen in the labile or stable cells at least. So we've done the physiological examples. So we've done breast tissue and liver cells. Also another example of physiological, if you're a mountain climber or if you're an athlete and you're constantly exposed to hypoxia through activity and exercise, your hematopoietic stem cells in your bone marrow would produce more and more red blood cells. So that's hyperplasia in your bone marrow to push red blood cells out to increase the oxygen carrying capability. Now, going to a pathological cause of hyperplasia, a good example would be the prostate gland. So it's exposed to levels of androgen hormones, and that causes the prostate gland to increase in size. So this is very common for males over the age of 50. So the prostate increases its size through hyperplasia, and that's in response to hormones, or in this case, androgens. Another good example of hyperplasia in a pathophysiological context would be if you're exposed to a particular virus, such as a papillomavirus. This causes, as a result, warts. So we have an increase in growth or hyperplasia in the skin, where you get these little bumps which are known as warts. That's caused by a virus, not in the case of a physiological cause. That would be considered pathological. So that's the atrophy, hypertrophy, and hyperplasia. So what I'll do now is I'll go to the last two. So I'll rub this out. And the last two are known as metaplasia, okay, and dysplasia. So with metaplasia, what's ultimately happening here is we're changing in the cell type or the morphology. So the way that the cell looks, it will change its type. Now this is usually in response to chronic irritation or inflammation. So when the environment of the cells are exposed chronically, so long term, to inflammation or a type of chemical irritation, the cells think, well, for us to survive, we need to change and equip our cells to be more like a different type of cell to survive. So this is an adaptation which is not changing the number, but actually the way it orientates itself to be more likely to survive. So if these cells were exposed to, say, a lower pH, like an acidic environment, it would want to produce cells that are more like the stomach, because the stomach has cells that are very good to survive a low pH. Or if you're exposed to chemicals like in cigarette smoking, instead of cells that have the cilia that are important to push stuck, particles in it with mucus out for a particular protective purpose, the chemicals in the smoke, the best type of cell to respond to that would be a stratified cell. So if we think about, imagine this is a ciliated cell, so it's got cilia on it. Okay, so this is in your bronchioles. Now, if you expose these cells to chemicals in cigarette smoking, instead of being a ciliated pseudostratified cell, it's going to become stratified squamous cells. So they'll become cells that will be many layers in thickness. Okay? So it responds in this way because it feels that this cell type is better when exposed to chemicals in the cigarette. So it can seem to respond and adapt better in this state rather than this state when it's responding to this particular chemical environment. It's important to note that in metaplasia, it is actually reversible. So this is metaplasia, and it is irreversible once you remove that stimulus away. In this case, it's going to be the cigarette smoke. Now, the other example would be in GORD, so gastroesophageal reflux disease. So in this case, we're starting off with cells which are stratified squamous cells in the esophagus. But as the stomach acid comes out of the stomach up into the esophagus, and these cells are exposed to acid, the cells actually respond by becoming more columnar-like, so more long like this. And the reason why it does that is because they're the cell types in the stomach, and it seems that this cell type does better when exposed to acid. So the take-home message with metaplasia is the genes that are activated are genes for differentiation. So the cells have to be not only a labile cell, but they have to be a stem cell, so a partially undifferentiated cell, so they can change their morphology to suit the change in environment. Okay, so that's important to know. But it's also important to note that the stem cell can't change to a completely different cell. So you can't go from an epithelial cell to a connective tissue cell or a muscle cell or a neuron cell. You can only be an epithelial cell, but the epithelial cell can change in its appearance from a stratified cell to a columnar or a cuboidal cell, or a columnar back to a stratified. Okay, so that's metaplasia. Finally, we go to dysplasia. Okay, so this is dysplasia, which means disordered growth. And in this case, what really happens is, again, the stimulus is going to be chronic irritation or inflammation, but unlike metaplasia, the response to this particular change in the environment is a deranged growth. So not only do we increase the number of cells, okay, so we increase the number, but we might increase the shape. So we might have flat ones, round ones, cuboidal ones, squamous ones, and it's going to be all disordered. So we have hyperplasia, so we have an increase in number, but we also have an increase in shape and the way that it's organized. So this is dysplasia. So the response from the normal tissue to dysplastic tissue is both an increase in cell number, an increase in cell size or decrease in cell size, and a way it's organized. So it's very disordered. Now, this is usually considered a precursor for cancer. So a good example would be in the cervix. So if imagine this is cervical tissue. Well, in cervical tissue is normally stratified tissue. Okay. So imagine that's stratified squamous tissue. Now, in this case, it's exposed to a virus, again the papillomavirus. After the exposure to the environment, it goes through a dysplastic growth, which makes it much disordered, and this is pushing it towards a precancerous state. So if you were to do a pap smear on the cervix, you would see that dysplastic growth, which is different to how it normally should look, and this is usually indication we're going towards a precancerous state. That is dysplasia, and that's why females are given the HPV vaccine, so the human papillomavirus vaccine, to stop that dysplastic change. Another example is in the bronchioles for preterm infants. So if an infant is born prematurely before 30 weeks, naturally for the baby to survive, it will need to be ventilated and given oxygen, etc. That exposure causes a dysplasia. So this is called bronchopulmonary dysplasia, and approximately 20 to 30% of infants born before 30 weeks will develop that kind of dysplasia. Now finally, unlike metaplasia, this can be reversible, but it's less likely. So this is really something to be aware of, because this is usually the precursor or the first stage of like a cancerous state. So hopefully now you've seen the different types of cell adaptation. So we can see a normal cell, and when it's exposed to changes in environments like workload, demand, hormones, inflammation, chemicals, growth factors, it adapts. It can adapt through getting smaller, getting bigger, increasing the number of cells, the morphology of the cells, and that's totally dictated by the cell type on how it essentially changes. But that adaptation is essentially to keep the normal cell alive. And hopefully now you can understand why they do it and how they do it.